For my NSERC undergraduate award this summer, I designed and carried out a study tracking the movement of European common blue butterflies. These butterflies are a recently introduced non-native species that is rapidly spreading throughout North America. Due to the fact that they're non-migratory, relatively slow flying, ubiquitous now in Montreal, and potentially invasive, we figured they'd be an ideal species for metapopulation movement studies. I'll talk more later about metapopulations, but for now, here are the details of the butterfly tracking. 3 or 4 of us, every day for 2 weeks, areas in a grid-like pattern around the MUHC hospital in Montreal. We split our efforts into 2 different types of tracking missions. One was a mark-release recapture experiment in which we would try to capture as many butterflies as possible within a randomly selected grid square given a time frame, usually around 5 minutes. Any butterflies caught would be marked with sequential unique identifiers, and their location and time of capture was recorded with the GPS. Any recaptures of those marked butterflies would give us information about their changes of location. The other tracking mission was a survey, which involved following individual butterflies for up to 10 minutes at a time, observing and recording their behaviors and flight trajectories. The photos show soccer cones, which were used to mark their locations during the tracking, after which they'd be recorded on a GPS. During the 10-minute track sessions, the butterflies' flight times, chosen food plants, interactions, and other behaviors were recorded with behavioral software on a tablet. In order to gather useful data for our purposes, during the surveys some special measurements needed to be made. Each time a butterfly took off from a spot, then landed. Here is step 1 and step 2. An alternative potential trajectory and landing site were also calculated, here as alternate step 1 and 2. These alternate steps were measured out with measuring tape and a compass using a realistic distribution of possible alternate flights the butterfly could have taken. Vegetation samples were completed at all landing points and all alternate points. A round square meter quadrat sampler was used for this. For each sample, an estimate of % coverage of the species' known food plants, flowering and non-flowering, was taken, along with any other significant details like dirt spots, other flowering plants, or concrete. So all told, we have complete and usable data from 18 surveys, and we captured and marked a total of 472 individuals. We recaptured 30 butterflies. 8 of those were recaptured on the same day, so counting only those that were recaptured at least a day after the first capture, our recapture total is 22. There were 2 butterflies that made it all the way from one side of the hospital grounds to the other. Number 103 was found 715 meters from where it was initially captured, 8 days later, and 68 was found 595 meters from where it was initially captured, 2 days later. So now that we have all this data, what's the plan? So as I mentioned earlier, the study was designed as an extension to an NSERC project I worked on last year. That project was the development of a metapopulation patch detection algorithm, Eigenpatch. Generally, metapopulations are models based on how animals move and organize on landscapes. Metapopulation models assume that organisms live and interact within patches, with rare dispersal occurring between patches. Specifically, Eigenpatch is looking to use animal movement models and random walk statistical models to better understand how animals might define patches. Using the data gathered from the butterfly study, the next step is to fit a movement model. The aim so far is to fit a spatial capture-recapture movement model using coarser movement information from marking, releasing, recapturing, bolstered with step selection information using finer movement information from the survey. We will then apply that movement model to the Eigenpatch algorithm to further test the algorithm's ability to detect patches. Thanks for listening to my NSERC USRA presentation. Thanks to Eric Pedersen, Emma Despland, and Eric Dexheimer. And thanks to Dario Di Girolamo, Natalie Dupont, and the other field volunteers.